Before we get into talking about what genetic material is made out of, let's talk a little bit about what genetic material is in the abstract. So for any form of life that we know of, one generation has to pass material or information to the next generation so that generation can build bodies for itself and survive in the environment. So a parent has to pass some sort of information on to its offspring. So the parent here is very broadly defined. Could be a human having babies, could be cats having kittens, it could be plants like, say, a flower producing more flowers, and it could also be bacteria multiplying, so splitting apart and creating more new bacteria. Some little baby bacteria right there. So very generally speaking, that's what genetic material is. It's the information that parents pass on to the offspring so that the offspring can make their own bodies and survive. Now, at the end of the 19th century, so in the 1870s and 1880s, there were a couple of really important experiments. So the first one of these really important developments was discovered by a guy by the name of Johann Miescher. And Miescher was a physician who was interested in pus because it was obviously really important for illness. And what Miescher ended up looking at were these cells that were found in pus that today we call lymphocytes or white blood cells. Now, one of the things about these lymphocytes is that they have these gigantic nucleuses relative to their size. So if that yellow part's the cell, the nucleus might be about that big. What Miescher developed was he developed a way to take the nucleus, the stuff inside the nucleus, and pull it out of the cell. He never got a whole lot because, as you can imagine, these cells are still pretty small. You don't get a whole lot of material out of them. But he was able to do some chemistry on the material that he was pulling out. So he's ended up with sort of the material that had been inside there like that. What he found was that it consisted of two things. First thing was proteins. He wasn't the first person to discover proteins. Proteins had been around. People knew about them. What he did discover was there this other stuff that behaved like an acid. And because it was coming from the nucleus, remember this stuff here is nuclear material, stuff from inside the nucleus, they called these nucleic acids. Now, around the same time, there was a zoologist by the name of Wilhelm Roux who was looking at dividing cells. So cells undergoing division. So by this time, people had noticed a lot of different sort of cellular machinery inside of cells. So let's say we got a cell here, and you've got a bunch of things. So these would be things like the endoplasmic reticulum and mitochondria, and you'd still have a nucleus in there, but this isn't necessarily a white blood cell, so maybe the nucleus is a little bit smaller relative to the rest of the cell. So what Roux noticed in his observations was that all this stuff divided pretty haphazardly. The endoplasmic reticulum would break up. The mitochondria didn't seem to be really ordered. What he did notice, though, was that the nucleus was very orderly when it divided. So what happens is you've got the cell, and eventually the envelope around the nucleus breaks down, and the things inside the nucleus go through this really highly ordered dance. You get this stuff that lines up in the middle first, and then you've got these sort of strings that they could see it even in the 19th century. They could see these sort of string-like things attaching to the well-ordered plate in the middle. And then each of the daughter cells, the two new cells after this old cell splits in half, they each end up with half of the stuff that was in the nucleus. So what Roux reasoned was that the stuff that was in the nucleus, because it was so ordered and because it was split in half the way that it was, had to be the stuff that made up the genetic material. Because the genetic material is the stuff that you'd have to be really, really careful with. Any sort of other machinery, if one cell gets 51% of it, the other gets 49% of it, it's probably not going to matter very much. But if you make a mistake with the genetic material, the instructions for actually living, cells would have a really big problem. It would be really hard to have life in a case where you're being disordered with your genetic material. And the only real candidate that Roux saw for a really ordered splitting apart in all of this was the nucleus. So if we take these two results, these Roos dividing cells and Meischer's observations in chemistry coming out of pus, if we take these two observations and put them together, what we get is that genetic material, which remember they still didn't know what it was, genetic material is protein and or these nucleic acid things.